So thank you, Kat, and the organizers for inviting me. This is an unbelievable session. And I know that the reason people are staying in their seats for such a long, late afternoon is because they're waiting for my 9-minute coverage of immuno-oncology. I thought I was just going to chair the session, and we have all-stars, but I always do what Kat says. So 9 minutes on immunotherapy. This is a slide that I borrowed from Steven Schoenberger at LJI, and illustrates the different types of immunotherapy with the prototypical T cell here, and then the different types of therapy, taking the brakes off with checkpoint inhibitors, the tumor-infiltrating lymphocytes. Is there a pointer or no? Maybe you can. Let me see. Tumor-infiltrating lymphocytes. So these, this is all being done. You know, this is Rosenberg at NIH is doing this, where it takes out a tumor and they amplify some of the cells and give it back to patients. And then, you know, there's a lot of activity here. I'm interested to see what Carl has to say. But the CAR T cells, which everyone knows about, and then now the new type of CAR T cell, I guess. And that's a TCR engineering T cell that's very specific and identifies more cells. And then this personalized vaccine approach, which I'll talk about in a few minutes. You know, I just wanted to show this because, again, this meeting was on cancer stem cells, or is on cancer stem cells and immune cells, and just want to show you how prolific the faculty are, you know, in the Mesa with elite institutions, LJI, Scripps, all sorts of institutions. And this list are cancer stem cell targets or immune targets that the study that proved that they were immune, at least in preclinical studies, are shown here on the left, and and all of these made it into the clinic within the past 5 years. And you know, it's just amazing to think about it. You know, I'm on EABs, and if they have one drug that they developed and just got to the clinic, it's hard to do. But it shows you sort of that entrepreneurial spirit that you know, Kat and Dennis and a lot of people have. So I'm not going to cover all these, but it's a pretty impressive list and a few FDA approvals. This is one that I just wanted to show. It's a little random, but it's not in the clinic. But I think it's an amazing finding by Kunlian and Dennis Carson that's showing that LATS deletion, which is part of the Hippo pathway, YAP deletion increases immunogenicity to the point where that the cells are killed in immune-competent mice, but not in immune-deficient mice. So it's a really totally different approach. And there's a lot of interest in biotech and so on in this. And then just to finish with some stuff that I do in this area, this is a figure that Ludmil will probably remember. And that is, if you look at intervention studying pancreatic ductal carcinoma, for a couple reasons. One of them is that it is the prototype of a really terrible prognosis disease, so that the 5-year survival has gone from 8% to 1 to 2% since 1940. And that hasn't been linear. And it also has very limited preventive and of course, therapeutic options. So, you know, lung cancer, smoking can affect it, and so on. There's nothing in terms of lifestyle that's been shown to affect this disease. And when the disease goes, these are just the pannins, premalignant lesions from Anavan Matra at Hopkins. When they transition to invasive disease, all hell breaks out. They have, you can see phenomenal changes in copy number, loss and gain, that happens almost at the same time as invasion. So that, together with everything I've told you, has brought this as an excellent model for prevention. And I have been doing prevention for longer than I want to think. I'm pretty sure I have more published negative trials than anyone else. I'm not sure if there's any other metric, but I've been doing it for a long time. And I think we fundamentally have approached it in the wrong way, and that the idea of using a cancer vaccine, not HPV, a cancer vaccine to induce in early premalignancy, we think has major advance. And so we actually just received a Stanford Cancer Award and using Liz Jaffe's KRAS model and showing activity, and a lot of really big players in the area, Jim Allison, Steve, and Craig and Nelson. This is just part of the team. You can see the teams and the team leaders below. Tyler Jacks is making new mouse models to be more useful in the immune setting. And this is one that I mentioned to you, I said that we'd get back to, and this is a novel approach called neoantigen calling workflow. I don't know if Steve is here, but he gave me permission to show this. This is, you know, now everyone's trying to use these fairly elaborate computational approaches to determine which neoantigens may be immunogenic or to target in any way, including vaccines. Unfortunately, I think it's safe to say, based on the recent Parker Institute analysis, that that has been negative, largely. So what was developed in Steve's lab, and we were collaborating, is this idea of using the algorithms to narrow down the neoantigens, and then, so that's the bioinformatic analysis part, and then to use immunologic tests on every neoantigen, so you don't assume anything. And you know when you do it that these are immunogenic. And we're looking at stool and so on. So it's pretty exciting. Steve talked to the Parker Institute recently, and they're really excited about doing this because, you know, this is a new way of doing it, and others haven't. So we're pretty excited about that.